Thank you so much, Dan. It's a real pleasure to be here. And I wanted to first start by thanking Matt and Jonathan Stamler and all of the community here for putting together this, not just the symposium, but just this amazing effort, which I think dovetails so well with the RTLP. So Dan invited me to give a few of my thoughts in a talk that I've titled Scientific, Regulatory, and Ethical Implications. And to introduce some of these ideas to you, I thought, I've had the privilege of serving as an advisor to the RTLP over the last few years. And my advice for this project has really stemmed from our personal experiences working with precision nucleic acid therapeutics. So in order to introduce some of these ideas, again, just to set the table for the discussion, which I think will be the most interesting part, I'll go through a few of the historical events that led us to this point. First, my disclosures. The concept that we've been talking about multiple times in this forum is the concept of using precision nucleic acid therapeutics as a method to greatly accelerate the pace at which we can deliver therapies. And if we can accelerate the pace at which we deliver these, we can conquer inequities that have arisen from our inability to make this fast enough to address the long tail of very, very rare disease. And many of the platforms that have been described with programmable potential, gene therapies, mRNAs, antisense oligonucleotides, siRNAs, CRISPR, folks ask, well, which one are you talking about? And the answer is really all of them. Of course, these types of approaches have amazing and exquisite specificity. They have the ability to target genes for upregulation or downregulation using a variety of different approaches: replacement, upregulation in situ, knockdown in situ. And increasingly, we can even now begin to responsibly talk about using them not just for gene targeting, but for mutation targeting. For very, very precise mutations that may be found in 10,000 individuals, may be found in 1,000 individuals, may be found in one individual. Of course, yesterday you already heard from Julia, who introduced this concept, describing an odyssey that we had the privilege of undertaking with her family as partners, where in 2017, 2018, 2019, we met her daughter, Mila, who had CLN7 Batten disease. And in the course of that 10-month sprint, we're able to recognize and conceive of a therapeutic opportunity. And then in collaboration with many individuals across industry, regulators, academia, we were able to bring to bear a patient-specific oligonucleotide treatment that showed significant benefit for her. To round out that scientific story, you heard how this treatment in the initial first year of coming on board elicited dose-dependent improvements in seizure frequency and severity, but ultimately was not a success in curtailing the ultimate outcome for this disease. It appeared that we had come too late to actually reverse the already ongoing brain shrinkage and atrophy that was occurring in this disease, and she did pass away in year 3. But to complete the story, one of Mila's family's enduring gifts, of the many that they continue to give to us, was the offering of partnership for donation of research specimens via brain donation. And through that gift, we were able to look at critical scientific elements to understand whether or not this intervention, which appeared to have clinical benefit, was also showing reflection of drug distribution and uptake and activity. And we were able to see cellular uptake throughout the brain using a personalized assay to detect this personalized drug. We were able to look at the distribution of a personalized antisense oligonucleotide drug delivered via spinal tap into the lower back. And we were able to look at this in a geographic way from the top of the brain to the base of the spinal cord and find that there was abundant distribution of melicin in multiple brain regions and spinal cord regions, corresponding to the expected biodistribution of what you would get from a lumbar spinal intrathecal injection. We were able to see as well that target engagement was elicited by this penetrance of drug within those tissues. That we could see rescue of our normal splicing pattern that we were hoping to get, that we had demonstrated in vitro before embarking on this clinical expedition. And we saw that it mapped very, very nicely in a linear fashion according to the distribution. And this concept of being able to move quickly and then deliver a drug using an existing platform that works, that gets where it's supposed to, and does what it's supposed to once it's in situ, really is the basis for this concept that we can now talk responsibly about trying to advance this programmable approach to many other conditions. In the 5 years since this effort was published, we have seen many other efforts like this to try and build up the body of evidence that this approach is appropriate. In our group, we've contributed, I think, these 4 programs involving 6 patients with the following orphan diseases: ataxia-telangiectasia, KCNT1 neonatal epilepsy, FLVCR1 retinitis pigmentosa. Each of these are conditions for which no previous genetic therapy, targeted therapy, had been developed, but now we have developed mutations, specific oligonucleotides that we've deployed for 1 or 2 patients, just a pittance, but 1 or 2 pilot patients that will inform the path for other patients with their disease and with others. And our work in this space is accompanied by many, many others who have now demonstrated this proof of concept multiple times, including from colleagues at Columbia University, at the University of Massachusetts. Colleagues of ours here in Europe are working to bring these to bear at the Dutch Center for RNA Therapeutics, and many, many other institutions as well. And LORM, the LORM Foundation, working in partnership with Ionis Pharmaceuticals. There are now approximately a dozen such cases out there that lay the path forward. Where might this path go? Well, we are thinking not just about gene-targeted therapies, but about therapeutically targetable mutations. This pie chart shows all 75,122 known pathogenic variants in the ClinVar database that show the distribution of the molecular types of variants that are clinically disease-associated. And to this, we were able to show that you can also add an additional slice of the pie that's not currently in the chart. These are gain-of-splice site variants that are deep within intronic regions that are treatable with splice-switching ASOs. The reason that these have been under-recognized in ClinVar is that there's been underrepresentation of whole genome sequencing within ClinVar, and these variants are only detected if you actually interrogate a whole genome, not just targeted capture. And we know that this is part, therefore, of the therapeutically addressable genome. And to that, we have ambitions to lay a foundation for then adding the types of variants that could be treatable with programmable CRISPR base editors or prime editing, such that now, in addition to gene targeting strategies, which are obviously the most efficient whenever they're available, that this category may also be someday available to help patients in need too. Now, important cautions in thinking about some of the ethical issues surrounding an ambitious proposal like this. This is, of course, the concept of repurposing drugs according to platform principles is simple. It's incredibly compelling, and it reaches out towards lots of folks who are currently underserved. But there also must be acknowledged that there are many hazards to navigate: clinical, regulatory, ethical, practical, and logistical. And as we do so, we are working with populations that have been neglected, and they may also they are also very vulnerable to these scientific opportunities and perhaps overburdened expectations. And so we have to be quite careful to communicate scientific possibility, but also uncertainty, and not to be overconfident as we talk about programs like RTLP. In doing so, though, patients always remind us that we can't be too negative. We have to weigh treatment risks against disease risks, and that's a careful balance that you'll see permeating through the thinking that's gone through the RTLP. In terms of interventional considerations, we're talking about conditions with compelling medical need, seriously debilitating or fatal orphan diseases, such that the risks of an experimental treatment are offset or at least need to be put in consideration against the risks of the condition itself, the natural history. We need to focus on single gene disorders for which genetically targeted treatments can be conceived and devised. And we are talking about conditions that are, by their nature, very rare, which means that it gives us the opportunity to do in-depth, individual, tailored consenting to ensure that the extra burdens of enrollment and participation in research trials like this are appropriately disclosed. This type of effort is now being addressed by multiple parties in the ecosystem, from regulators to academics to collaboratives. And the RTLP is joining a fantastic group of folks, but the RTLP has some very special superpowers that distinguish it from many of the activities in this group. Before getting to the RTLP, however, I would like to call out that recognizing the need to address not just scientific issues, but these regulatory, ethical, and logistical, and sometimes even business challenges. We had put out a call to the community to gather folks who wanted to sit and work and wrestle with us with these issues via the N of 1 Collaborative. And this is an independent coalition, a nonprofit that we started some 3 years ago that's committed to developing repeatable, rigorous, and responsible pathways for individualized medicine. And 3 of our goals are outlined on the slide: to connect patients and scientists, to open source the process of making programmable medicines, and to train the next generation of interventional geneticists. What this group lacks, however, it has intellectual authority, but not operational authority. And I think that that's one of the really unique advantages of the group that's gathered here in this room, and also over the last year and a half of planning for the RTLP, that there's a unique coalition of expert scientists with folks who also have the authority to make decisions within a much more coherent system than exists in my country. And with this group, I think that some of the challenges and opportunities that will be discussed and will be very carefully planned for are listed on this slide. How do you rigorously establish efficacy for N of 1 drugs? What are the tools that you use, the clinical trial design tools or single case study design tools that you use to accomplish that? Basket and umbrella trials, master clinical protocols, adaptive designs. What roles for surrogate markers that might be used to accelerate the determination of efficacy in a patient might be appropriate to deploy if you can't wait, say, 3 years for a positive neurodevelopmental outcome? And how do we actually coordinate at a human and professional level iterative learning via a prospective registry and database to capture the learnings from these cases? Number 2, how do you make programmable drug development more efficient? How do we provide appropriate, proportionate, yet also streamlined safety testing that reflects the commonalities as well as the differences from drug to drug within a platform? How do we manage the logistics of delivering these just-in-time drugs, perhaps with just-in-time small-scale GMP manufacturing? And how do we build our solutions to these questions in a way that will lay the groundwork for all of the different types of platforms that we conceive of using, the nucleic acid precision therapies, not just ASOs, but siRNAs, CRISPR, et cetera. Last, how do we develop this via a process that's transparent and open that allows for fair and judicious access? What are the standards by which a given effort should receive a regulatory stamp of approval sufficient to proceed at a particular velocity? What are the standards by which a given effort ought to be reimbursed so that this can be a sustaining enterprise? There are very interesting questions about whether work at this level, at the gene level, or increasingly at the mutation-specific level, is treatment or research, and patients and ethicists and physicians may have different perspectives on this. But the existing frameworks that we have for clinical trials, compassionate use, clinical innovative care, those are some of the tools and the vocabulary that we have at our disposal, but are they fit for purpose? And I think there are many ways in which they're not. Finally, just at a practical level, at the end of the day, what is the product that comes from a platform like this? Is it a marketed drug that for particular indications with a label? Is it a platform approval for a process, like an individualized cancer vaccine? Or is it an interventional procedure, like the way that we manage complex therapies in a multidisciplinary fashion, such as organ transplant, which is the intersection of medical expertise, complex and impressive immunosuppressants, and surgical know-how. I leave these as the questions and provocative questions for discussion in the panel, and I'll just stop and thank our team members and many of the folks in the community who have done the work that I've briefly described, and I look forward to questions and the conversation.